All right, in this video, we're going to be going over part 2 of Unit 2, Cell Structure and Function, for AP Biology review for the AP exam. My name is Mr. Chipman, high school teacher at Murray High School in Murray, Kentucky, and hopefully I can help you with this. Hopefully you'll take an opportunity to look at my other review videos as well. I spent quite a bit of time making these videos. Hopefully they're helpful to you. If you like them, please like and subscribe to this channel. I would very much appreciate that. And tell others how you liked this and show them the video as well. Hopefully it can help you, not just today, but even if for some of you who continue your biology careers at university, you can continue to use these videos as reminders. So let's go into the second part of cell structure and function, which is primarily going to be dealing with membrane permeability and concentration gradients and all the like. What is a concentration gradient? Concentration gradient is simply when you have basically two areas. In this case, we're going to be talking about the inside and the outside of a membrane. And one of those areas has more concentration than the other area. In this case, we have blue dots and not blue dots, right? And so there's a gradient, meaning that there's a high concentration on one side and a low concentration on the other side. If those materials are able to pass through the membrane, they will, until those gradients, or till that concentration equalizes on both sides, or until what we say that concentration is at equilibrium, meaning that both sides are equal, or that the particles are moving across the membrane at an equal rate, is another way of thinking about that. Well, the movement of those particles across a semi-permeable membrane that does not require energy is called passive transport. Again, the key element here for passive transport is that transportation is happening without the use of energy. The movement of those particles from a high concentration gradient to a low from a high concentration to a low concentration is just simply called diffusion. This is the movement of any particle from a high concentration to a low concentration. The movement of particles from a high concentration to a low concentration using some sort of other vessel, like a protein, is called facilitated diffusion, because facilitated diffusion means that there's something facilitating that diffusion, something is necessary, and it's going to usually be some sort of protein vessel. We'll probably talk a little bit about that in a moment. Active transport is the opposite. This is where you're going literally against the gradient. So you're going from an area of low concentration to an area of high concentration. The way that I like to describe this is, imagine you have a closet that's crammed full of stuff, so much so that it's just ready to open. And as soon as you even touch the doorknob, all of it's going to spill onto you. The act of that stuff spilling onto you is passive. You did not have to apply energy to get those things to tumble out of your closet. In fact, that energy was stored up. And so cells will oftentimes use concentration gradients in order to do some sort of cellular work. Well, the opposite is true with active transport. Active transport is like you taking all those things, your mom tells you to clean your room, so you're taking all those things and you're shoving them into your closet, which requires energy. It requires energy to shut your closet door. In fact, it requires a lot of energy. And you store all that energy up so that the next person that goes to open your closet, all of that energy is going to be released onto them. And so that's what cells create gradients in order to use them in other capacities. Another type of transport that we'll talk about in AP Bio is the transport of large molecules. Molecules that are so large they can't simply just pass through the membrane, or even they can't pass through some sort of facilitated diffusion, but they require either endocytosis, which is the taking in of large molecules. You'll either see this called pinocytosis for liquids or phagocytosis for solids. You can see here, what happens is basically the membrane will kind of envelop that particle and break a portion of itself off, creating a kind of temporary vacuole or vesicle in this case. Sometimes it's called receptor-mediated endocytosis, which is basically where a particular receptor is being triggered in order to trigger the process of endocytosis. In this case, you can see these little green receptors are looking for stars, and when they see them, they facilitate endocytosis. The opposite of this, of course, is exocytosis, where a vesicle is fused with the cell membrane and pushes those particles out of the cell. This could be to get rid of waste. It could be to put a hormone out into the extracellular space. There's lots of reasons that exocytosis could occur. So here again is facilitated diffusion. Facilitated diffusion is anytime diffusion is happening with some sort of protein, either a protein channel or a carrier protein. Either one of those would be an example of facilitated diffusion. As long as, again, diffusion is requiring no energy. Sometimes you'll see something like a protein pump. And a pump, by definition, if you think about a bicycle pump, it doesn't just sit there and do anything. It requires you to input energy. So when we're talking about protein pumps, we're not talking about facilitated diffusion, but we're talking about active transport. So here's facilitated diffusion again. This is an example of how different ways it can happen. So some things just go through on their own. We call those uniport. Symport is where one particle needs another one in order to kind of push it through. These are called co-transporter. They're sharing the transport. And in antiport, they're going in opposite directions. So the symport means together, anti means opposite. So they're just using, in order to do facilitated diffusion, they need another particle. So let's talk about this idea of membrane potential. We've kind of hinted at it a little bit. This is the potential of a membrane to do work. And so cells will use membrane potential in order to do some kind of cellular work. It could be an electric potential that's built up, like in the case of a nerve cell, or just a simple concentration gradient that's built up. The high concentration has a potential to do something because it's high and it wants to move to the low concentration, just like the stuff crammed into your closet has the potential to do something. All right, and this sodium-potassium pump is an example of how that membrane potential is arrived at. The sodium-potassium pump pumps two things, sodium and potassium, and it pumps sodium into the cell and potassium out of the cell. And what it's doing is it's building concentration gradients so that it can do work at a later time. It's building a membrane potential so that that membrane potential can do some kind of work. It resets the nerve cell in this case. This is just an example that you're given. It used to be content in the AP Bio, but now it's just typically an example that is used. Anything to establish a gradient is going to be called a pump. Osmosis, moving on to osmosis. Osmosis is the diffusion of water. So it's the passive transport of water. A lot of times when we think of diffusion or we think of passive transport, we think of materials like these purple dots. Well, the water actually moves too. And so we have a semi-permeable membrane here. This membrane does not allow the passage of materials. And so water is able to pass through it, and so the purple dots can't pass through it, and so water is at a high concentration here on the left, right, because there's more water here than there is purple. And over here you have a lower concentration of water, so water wants to move from high concentration to a low concentration, and that's exactly what it does. It moves and it creates until it's at equilibrium, just like the particles would do if they were allowed to pass through the membrane. Sometimes you'll see this called osmolarity as well, associated with this, and it has to do with a solution's potential to do work, and this has the directly involved with the amount of solute that is in that potential. Solute in the solution. Solute is the dissolved substance in the solution, and the solvent is the dissolver, usually water. Solute would be like if you made salt water, salt would be the solute, and solvent would be water. And this brings us to this concept of tonicity, which is a very important concept in AP biology. Tonicity has to do with the comparison of the amount of solute. And so you three words that you'll see associated with this are hypertonic, hypotonic, and isotonic. You never see these words by themselves. They're always words of comparison. And so a hypotonic solution means that something else is hypertonic to that, right? And so if this solution is hypotonic, what that's saying is that there's less solute in that solution than in something else. In this case, it's a balloon. And so, for instance, if there's more solute inside the balloon than outside the balloon, we would say that the balloon is hypertonic and the solution is hypotonic because there's less solute, or you could look at it as more water. Well, if there's more water outside the balloon than there is inside, what is water going to want to do? It's going to go in the balloon and cause the balloon to swell. The opposite is the case if the balloon is in a hypertonic solution. So let's say you have a balloon that's full of fresh water and the solution is salt water, then all the water is going to want to go out of the balloon, causing the balloon to shrink. And it's going to move until those solutions are isotonic, meaning that there's an equal amount of solution where there's no net flow of water. In plant cells, this can happen. Same words here. In a hypotonic solution, this causes the plant to become turgid, and turgid just means that the cell is so full of water that it's pressing against the actual cell wall, and this is good for a plant because it causes the plant to stand up nice and straight and point itself to the sun, do photosynthesis. At isotonic, it's called flaccid, meaning the plant is kind of wilted and that can need water. There's no net movement of water. And what can happen in a hypertonic solution is that all the water can move out of the plant, called plasmolysis. This is actually where the cell membrane tears away from the cell wall. In animal cells, since there's no cell wall, if you put an animal cell into a hypotonic solution, it will burst. All the water will go into the cell and cause the cell to burst. In an isotonic solution, there's no net movement of water. But if you put it in a hypertonic solution, that cell will shrivel. This is why Grandma said to put salt onto slugs, because all the water will come out of the slug and kill it. Don't do that to slugs. And this brings us to the concept of water potential. Water potential is just simply water's potential to move and to do work. When we have water potential problems, they're always going to be dealing with two situations. There's going to be some kind of cellular membrane or some sort of semi-permeable membrane, and what you're going to be asked to do is determine which way water moves. And water is always going to move from a high water potential to a low water potential. Up here, one thing I didn't say, I want to go back and say it. Water always moves from hypotonic to hypertonic. So if the cell's in a hypotonic solution, water's going to move to hypertonic. So hypo to hyper, O to er. So I always say it. And for water potential, water always moves from a high water potential to a low water potential. Well, how do you solve water potential? Water potential is the pressure potential plus the solute potential. And you're going to do this for both sides of the system that you're talking about. The pressure potential is always going to be given in a problem. The solute potential, more than likely, you're going to be asked to solve for it. Pressure potential is like how much pressure is acting on that system. It could be a positive pressure or it could be a negative pressure. Negative pressure would be like a plunger. Positive pressure would be like pushing down on a system, like a piston or something. Whereas solute potential has to do with the amount of solute in that fluid. And because solute's always trying to pull water toward it, water always goes from high to low, right? So solute potential is always trying to pull water toward it, meaning it's always going to be low. Solute potential is always going to be a negative number every single time. So you add those two together, you get total water potential. Well, how do you get solute potential? I have a question, or I have a formula here. This is a formula to be provided on the AP Bio formula sheet, and this is how you solve it. It starts with a negative every time, so don't leave that out. A lot of students miss it because they leave that negative off. I is the ionization constant, or how many particles of molecule are made in water. It's usually going to be 1 or 2. 1 is going to be something like a sugar, like glucose or sucrose, and 2 is going to be like your sodium chloride or hydrochloric acid or sodium hydroxide. Acids, bases, salts are usually going to ionize into 2 particles, whereas sugars are just going to ionize and stay 1 particle. Molar concentration, you don't have to know how to solve for molar concentration in this class, so this will be given. Pressure constant is always given, it's the same number. And then temperature will probably be given to you in Celsius, and you add 273 to it. Again, this is something that you're given on the AP Bio exam formula sheet. And you're going to be given this for one side or the other. Both sides are going to have, so I'll see if I can go back to this problem up here. And so this is showing water potential of a pure water 0, no pressure acting on it, no solute. Of a dilute solution, meaning that very little solute is negative 200, and a very concentrated, negative 500. So water wants to go from negative 2 to negative 5, high to low, right? A lot of students forget their number line when it comes to these kinds of problems, so make sure that you're paying close attention to this. And this has a big deal on osmoregulation, which is the regulation of the amount of water in an organism. Organisms are constantly trying to maintain the amount of water in themselves, keeping that at normal homeostatic levels, and so they have to manipulate the amount of solute in their bodies and the amount of water in their bodies. A lot of organisms, like more complicated organisms like a fish or like humans, we have kidneys to help us do this. but simple organisms will just do this through their skin, through just basic osmosis and basic diffusion through their environments. And so they will typically be equal with their environments a lot of times to help them keep down on the amount of net energy that is necessary to do that. A lot of times you'll see systems with these water potential problems. You will see, and the bell's about to ring, but I'll finish up. You'll see problems that look like this, these systems, and it'll give you the water potential on one side, pure water, 0, no pressure, no solute. Over here you have no pressure either, but a negative solute potential, and water goes from left to right. Very simple idea. The big problem with this is solving for solute potential. You'll be given that equation, so it's not all that difficult. Hopefully this was helpful. Please like my videos, subscribe to my channel, more videos to come. Thank you for watching.